This video will show some of Phenom-Networks' capabilities regarding correlation between continuous traits, their statistical analysis, and graphical representation. The main analysis to correlate between continuous traits is the XY plot. After I select this analysis, let's select one or more studies from which I'll take traits for the correlation. And I'll pick this one. All traits for the selected study appear now on the list. Using the Traits Display window, I can select traits by groups. Here I'll select the morphology group of traits. And now only morphology traits appear on the list. Now let's correlate between the traits bricks as the X variable, and then total yield as Y. Also, I'll put fruit weight in the color field and the variety name as label. The result shows the correlation between the X and Y traits. A data point's color indicates its fruit weight, such that green is low fruit weight, black is average, and red is high. When we point on a data point, we can see its variety name. On the top, there are the correlation parameters, such that R is the coefficient, N is the sample size, or in other words, number of data points, and P is the p-value. The plot's title shows all traits that are involved in the analysis. Varieties that were measured in this study can be divided into two categories. To reflect that in the analysis, we'll use the split by option. Then put the germplasm category in the by field. This vector provides the category of varieties. Then click on OK to repeat the analysis. Now we get a figure for each of the two categories. This is the figure for the first one, and this is for the second. We can see that the direction of the correlation is inverted between the categories. The first is positive, and the second is a negative correlation. In this study, each variety was tested in several replications. So each data point in the current analysis represents the mean of all these replications. Now suppose that we want to display all replications and calculate the correlation based on that. To do this, I go back to the Form tab and click on the Summarize by GID checkbox. This checkbox is responsible for summarizing all replicated data. When we remove it, all replications will be considered for the analysis. Moreover, because now we'll have several data points for each variety, we may want to put the replication name in the label field to distinguish between several points of the same variety. So let's put this factor over here. Each plot now contains many more data points that represent all replications. We can also see that the sample size is higher than before. When we hover over each data point, we can see the name of the variety and the replication. Some data points that are outliers are labeled automatically by the system. I can put several traits in the color field. Let's remove this one, and then, for example, let's put these two. This one and this trait. Put them in the color field. Now data points appear as boxes. Each box is comprised of two little squares that represent the two traits that I put in the color field. Traits composition of each box is described on the bottom legend. such that the left square represents green shoulders and the right is leaf veins. The color's values are given on the top legend, for each trait separately. When we point on a box, we can see the actual trait values. For example, this one has green shoulders equals 1, and also leaf veins equals 1. For this box, the values are 3 and 2 respectively. Gray squares indicate that no measurement is available. Similarly, we can put more traits in the color field. For example, let's select these two and add them to the field. The boxes will be split into more squares. Finally, as is true for most analyses, I can select data points from the figure and move them into the list page so they can be grouped together in a list. The XY plot 2 is very similar to the previous analysis, except that it provides a better possibility to split the results. To demonstrate it, I'll do the same correlation as before, with bricks as the X variable, total yield as the Y, and fruit weight in the color by field. I'll disable the Summarize checkbox to see all replicates. Then there's the Split by field, which can take up to 2 traits and is used to split the figure into distinct panels. Let's put into it the trait fruit size, and then germplasm category. And this is the resulting figure. It is comprised of several panels according to the traits we put into the split field. There is a panel for each combination of levels. The top row represents the ID level of the first trait, and each of its panels corresponds to a different level of the second trait, which ranges from 1 to 6. On the bottom there is the next level of the first trait, and again all levels of the second trait. On the top left of each panel there's the correlation parameters that were calculated for the specific panel. We can click on each panel to zoom in on these particular levels. For example, let's click on this panel. The correlation of these levels are now plotted as a separate figure. Of course, the correlation parameters are the same as were written for the original panel. The traits pairwise analysis is a simple way to create scatter plots from multiple traits and display them as a matrix. Let's take all continuous or ordinal traits and put them in the Y field. Then the variety name is in X. Again, we can color data points according to any trait. Let's pick this one. The figure shows scatter plots among all selected traits. On the diagonal there is a distribution figure for each trait. On the bottom triangle there are all correlation plots of the corresponding pair of traits. The upper triangle provides the correlation coefficient. All analyses that I showed on the tomato database can be performed just the same on any database and on any organism. For instance, let's do some analyses on rice. For this, I select rice from the organism menu to move into there. And this is the analysis page of rice. It looks just the same as the tomato, only that it contains rice data. Now, to do analysis, let's select Studies. I'll pick this folder, and from here, let's take all studies since the year 2000. If I click the plus sign in each trait, I can see all studies that this trait was actually measured in out of all the studies that I've just selected. Different traits may exist in different studies. Now I'll select the XY plot 2 analysis. Now I can put traits into the fields just the same as I did in the tomato database. This analysis will be performed on the combined dataset that is comprised of all studies that I've selected. So let's put flowering as X variable, and then grain yield as Y, and let's put plant height in the color field. Also, let's put leaf blast in the split field. And this is the resulting figure that describes the selected traits for all rice genotypes since the year 2000. Yet, as another example, I'll go to the mouse database. Here I'll select the folder of behavior studies. Selecting a folder means that I select all studies within it. These studies are comprised of several traits that were measured on the same mouse population by independent investigators. Let's select the traits pairwise analysis, and here I can put several traits into the Y field, variety name as the X, and I can also color it according to a trait. And then OK. And this is the resulting figure that shows pairwise correlation among all selected traits. Note that here a data point represents a mouse instead of a plant, as in the tomato and rice databases. Other than that, the concept and the analysis are performed and displayed just the same.